the system, the PlantArray system. I can say that in the last 2 months, 6 different papers were published. So this is accelerating over time. I will discuss with you later on about different applications of the system. And I also put here some names that you can see the customer types we have and some of our distinguished customers. Wageningen University, I believe everybody knows them. Göttingen from Germany. Korea from a chain of research in Italy, research institutes. And Florida University from the US. All are very well-known organizations in the research in the scientific arena. Mars Wrigley is a representative here for different customers we have in the food and seed businesses. Of course, this is intended for their R&D. And ProFarm is the representative of the chemical and biostimulants or fertigation type of companies that we have in our portfolio. So this is just to give you a bit of a feel who our customers are. And this is about the company. Here you can see the system itself. This is the system that we have installed at Wageningen University. In Wageningen, it's in their phenotyping, new phenotyping facility, the most advanced phenotyping facility in the Netherlands. And you can see that the system is actually being used for different applications, different type of plants. This specific picture is taken from their quinoa breeding program, which was sold to a commercial company, Radical Crops. So this is just one example of very practical use also for the research that was done there on the system. Let's talk about the principle of the system. So the principle is very much in our mind like a smartwatch. It works in the very similar directions. It is a dynamic measurement of different physiological parameters. So very much similar to that, to the smartwatch, and you can have this implemented not only for one person like we have in smartwatch, so not only for one plant, but for many plants. And what we do a comparative analysis of dynamic physiological measurements between the plants. In this, you can see the 3 functions that we have in the system. It's the control function, where we can control each and every plant personally with its own protocol. We control the water and soil conditions, so water regimes and fertigation. And this is you can have a feedback control, which is based on different parameters we're measuring or different sensors we are using, and then also maintaining throughout the growth of the plant the settings that you set in the beginning of the experiments. Of course, this is done by the software. And we measure the plant physiology together with the dynamic environment as well. At the same time, we're taking about 500 times measurements a day. And all of this goes together with different sensors into analysis. So the readings and also the calculations go into the analysis software, which is doing all that in real time, including statistics. Because all of the traits we are taking are absolute quantitative traits, absolute measurements. So what does it do, all this? It actually gives us better understanding of the plant-environment interaction. This is what it provides the researcher, the ability to have deep insight into these interactions, and also to save time in the process. Instead of full seasons, trial and error in the field, you can now do very much controlled, in a controlled environment, you can do within a few days, get a very good understanding of where you're going with your plants or with the materials. How we do it and how this magic actually happens is that we measure the traits that we're measuring are physiological traits. And these traits, for example, here you can see transpiration and stomatal activity, but these two traits, and these are just examples, we have more, they are correlating nicely with the yield. You can see the linear correlation between transpiration and grain yield. By the way, this is taken from papers published. So based on that, within days, you can actually understand between the comparing different plants, which have the best potential yield between them based on these traits that we are measuring. So what are the traits? You can see here in green are the plant traits, and in blue are the environmental traits or parameters we're taking. You can see with the technology we're measuring the transpiration. This is on a dynamic way, about 500 times a day. Biomass gain, which is the wet biomass of the whole plant during the growth of the plant once a day. And based on that, we can extract the water use efficiency and the stomatal conductance. These are all direct measurements being taken with the help of a mini scale. You can see it here, a lysimeter, which is underneath the pot. And this helps us to directly measure those parameters. And also we are measuring root influx, which means the root water uptake. So it's the root activity. Together with all of these traits, we are measuring the atmosphere. You can add different sensors, or either for the atmosphere or for the soil sensing or for fertigation in the reservoir, or even additional plant sensors like NDVI and temperature. All of this goes into a cloud software where it's all being analyzed in real time, again, because these are absolute values being taken by these scales. This is the controller which we have for each and every pot. This helps us to do personalized treatments to each and every pot, and also to provide multi-solutions treatments. So you can see in these examples, A and B meaning two different independent solutions. You can have more than two. And you can control it by many different ways, either by time or by feedback from the weight, from the scale measurement, or from the different sensors like the soil sensing or any other sensors, or even by the own plant activity that you are measuring. You can control by, for example, by the transpiration of the plant. It has different advantages. I will not go now into that, but you can see here the versatility of the many different ways how you can control, and this is the screen from the software, how you can control the different fertigation lines. Here are just the different applications that you can apply with the system, the research for, so genetic screening, abiotic stress studies, different abiotic stress studies. If you put the system also in a controlled environment in terms of atmosphere, you can even split it between two rooms, no problem. It's the same system. You design the experiment through there within one system. Nutrients and bioag in combination with stress studies. Biotic stresses of root and stomata activity, we found that there is an ability to early detect much faster from any other devices the stress by the plant. Seed treatments, how they're impacting the whole plant, and also growth practices optimization like irrigation practices, soil optimization, etc. The system works with many different plant types. We don't have any limitations so far, from Arabidopsis, very small plants, until plants that are, even trees that are quite, let's say, young trees or fruit trees can be also grown on the system. How are we actually different from other high-throughput devices? The main high-throughput device you can find out there, it's imaging. And how are we differentiated from imaging? Is that we are the main, let's say, the main differentiator is that we are measuring the activity of the plant, where in imaging you are measuring mainly structure or color. The change of that. So we are measuring the actual function. It's, again, I remind you, the smartwatch. This is more or less that. Then we also measured the plants simultaneously. All the plants, if it will be 500 plants, all of them are measured simultaneously. At the same moment, we take a measurement from all the plants, and we take them also continuously, continuously dynamically, where in the imaging, you can do only a few plants at a time or few data points a day. Also, the analysis is being immediate because the values we are measuring are absolute and direct. And in the imaging, it's usually complex analysis. You have to analyze cloud points and translate them into quantitative traits. And in our case, it's very easy also in terms of data storage. And you don't need to hold and maintain servers. Also, we have this controller that I described with flexible personalized controller, where in imaging, usually it's very limited to a group of plants, and also quite limited in what it can do in terms of the flexibility. So that's it just for the overview, and after me, Menachem will talk about the different applications or use cases, real use cases of the system. You can raise questions in the chat, and we will try to do our best to answer within the short time. But if not enough time, you can also write to me. This is my email. Feel free to write to me as well. So thank you for your attention. I will just now stop sharing, and I will let Menachem take over. Please, Menachem.
Okay. Thank you, Karen and Laura, for this nice introduction. Let me also share a presentation. Okay. Do you see my presentation?
Yes.
Okay, good.
But yeah.
So, hello everyone. So my name is Menachem Oshelion, as I mentioned before, and I'm a plant physiologist from the Hebrew University. I collected few samples to show you how we can use the PlantArray system in our research. My example going to focus on 3 main points. One is breeding. This will be the main. And the other one will be how you can actually detect a cellular mechanism or a gene-related mechanism. And the third one, if we have time, I'm going to talk about biostimulant screening. So the first research hypothesis I'm going to present is breeding. So I guess you know that drought breeding or breeding for improved drought response may take years. In this research, what we did, we made the hypothesis that we can maybe do a
Menachem, you unmuted yourself. Please.
Sorry, can you hear me now?
Yes, we can hear you. Sorry.
Okay.
For
Sorry.
the interruption.
So our research hypothesis here was that if we can take data from field plants that we already know that was bred for drought, and then do a reversed phenotyping on the PlantArray system, maybe we can understand cellular and all plant physiological responses. And by this understanding, maybe we can improve our future screenings. And this is what we did. We took Dani Zamir's
Menachem, again, you have switched off your microphone.
Oh, how is this? Now it's okay?
Yes, now it works. Thank you.
I don't know what happened. Okay, never mind. I hope it won't happen again. So we work with Dani Zamir's tomato interrogation line. Just in short, to those of you who doesn't know, Dani Zamir actually crossed the wild-type Pinelli with the breadline M82, and then did backcross, leaving the M82 almost the full genome, but a small piece coming from the father, the Pinelli. So these lines are very similar to the mother plant, the M82, but with a small piece of a genome in each one of the chromosomes. Dani Zamir was generous enough to give us a lot of data coming from many years of field research. So we took all the field research of all this line, and we looked for a few years of research where we compare always the M82. The M82 is the control plant. And we, from all the 76 different lines, we actually choose 30 that showed either better field performance, and I'm emphasizing this. We work on the field performance. So for example, if this line 1211 has higher fruit in the field and higher shoot weight, also under drought conditions, so we call it high, high, high. When I say high, it's at least 20% more. And this plant was selected as idiotype. And so on and so forth. Of course, it's very hard to work with all of these, so we choose few that will be either idiotypes or good plants or good performing plants or bad ones. The most important thing that we choose only plants with at least 4 years of field experience. So we didn't want just to select some plants, we choose few. I'm not going to get into too much details, but let's focus here on one of the idiotype 5.2, this plant, and one of the bad plants, 18.1, this one. So we put all the plants on the plant array system. We build a randomized structure where each plant was randomly placed. We had at least 4 to 5 independent biological repetitions, and we measured the root, the plant, and the atmosphere, what we call the SPAC, soil plant atmosphere continuum. As Karen says, all the data go to the cloud, and we monitor, you see, continuously the atmospheric condition, light and VPD, the transpiration, where each line you see here, each color is different plant. We monitor it for control, and then drought, and then recovery. All took about 1 month. Okay, so this is the experiment. Let's see some result. Here you see, let's call it the bad line, the 18.1. You can see what Karen called previously functional phenotyping. What do I mean by functional? It's the daily transpiration day by day. Day 1, and this is day about 20. You can see actually the plant is growing, which means more and more transpiration, which means more leaves. When you put next to it the 5.2, they grow together. Remember, all of them was growing simultaneously on the same platform, like you see here. You see this 5.2 at the beginning was the same, but very quickly it developed much faster. So we can say 5.2 has much better vigor, and you can see it statistically already from day 6 or 7. These are the rest of the line, but as I said, let's focus only on few. So on the pre-stress, you can see very clear differences, very fast differences, and you get significant relatively fast, much before you can see anything with your eyes. The plants look very similar at this time. Then we want to expose them to drought. So in B, you can see the soil water content. This comes from the soil sensors. You can see no different. All of them are in the high water capacity. But now when you want to expose plant to drought, this is a big bias. I want to show you this figure. If you just close the tab, so for example, if we close the tab here, you will get some plants drying very fast, like you see here, and some plants drying very slow, like you see here. All of these plants you see here are the introgression line population. So just closing the tab is not a good idea because you're going to get very big different treatments. So these plants, for example, will be under stress after a few days, while this one won't be under stress at all. So drought treatment, a good drought treatment, cannot just close the tab. What you have to do is to compare apples to apples, or to expose all the plant to the same stress level. We did it using the controller of PlantArray, as Karen explained before, and what we use is the dehydration rate. Dehydration rate actually fixed a steady drought scenario to each of the plant based on the plant transpiration. So it doesn't matter if one plant transpire a lot and another one transpire less, they will get an equivalent amount of water. The high transpiration will get more than the low transpiration to keep all of them, as you can see here, in a steady drought condition. Doesn't matter if the plant transpire a lot or not. This is the soil water content. So in the soil, they get similar drought. So of course, the air, they have the same, they're exposed to the same light, VPD, and so on. But also the drought is similar. This is only possible with a feedback irrigation system like the PlantArray. And then we put them in recovery. You can see the recovery here after the stress ends. We decided to recover all of them. They recovered at the same time. And you can see, for example, this 5.2 line, the isotype recover much faster than the 8.1. And this is very interesting because the 8.1 did not transpire so much. And even though it recovered less than the 5.2 that transpire a lot, exposed to drought, and then even recover faster. So another thing we want to test is the stress itself. Under stress, you can see that the plants are exposed to pretty much they close the stomata. You can see it here. But if we really want to measure, compare the plants under drought, what can we do? So of course, measuring the plant at this position, here I took only these two lines, the 5.2, the idiotype, and the 18.1, the bed plant. So you can see that under stress, you don't see any difference. It's here. But at the recovery, the delta between the control line and the recovered show you that the 5.2 recover much better than the 18.1. And this is a very nice way to look on the recovery because the recovery actually reflect what the plants did under stress. If the plant could maintain good survival mechanism, it will recover much faster than this plant that recover much slower. But additionally, you also want to analyze what happened here under stress and compare the plant here under stress. But as you can see, they don't have too much difference in the whole day. This is the daily transpiration, which means how many ml of water the plant loses in a day. So what we want to take is take the plants to much better resolution and look at the momentarily resolution. You can see this is 1 day. So every minute we can follow the light, as you can see here, the VPD, as you can see here, and also the plant transpiration in gram per minute. So the blue line gives us transpiration minute by minute, and you can see the plant reach its peak somewhere at noon and then close stomata again. So this is really water flux. But the system can also give us, and this is the water in the soil. So of course the plants take the water in the soil and just transpire them to the atmosphere. But the system can also give us the stomatal conductance, which means what is the aperture of the stomata. And here you see a very different pattern from the water flux. So the stomatal conductance show you that the stomata is much more open early in the morning or around here, when the light is around 600 watt per meter, which is very nice, but the VPD is low. So the plant is much more efficient at this time of the day than at noon. At noon, the stomata is closed, and because the VPD is high, you lose much more water. So these two very interesting functional traits are only possible because the system has a very good signal-to-noise ratio. I will explain it in a minute. But now you want to measure this part at this point, at the drought point, okay? How do you know that you really compare the plants to the same drought level? So what the system gives us is the point of the stress. It's called theta crit. Only from this point on, the plant is under stress. So we measure this point to each and every plant, and then we take 50% from this point. 50% of the theta crit is actually for us the same stress level. So you can see we measure the stomatal conductance. This is actually reflect how much CO2 the plant's entering through its stomata. And this is the 18.1, the bed plants. You can see this is the pattern of the stomatal conductance, and this is the transpiration pattern. But now when you look on the 5.2, the idiotype plant, you see a very interesting performance. You can see that the plant is much more active in this, what we call the golden hour. This delta is very important because at this time, the plant almost do not lose water. Remember, the VPD at this time is very low or relatively low. So it's a very smart strategy to open stomata early in the morning, and you can see you lose less water at this time. This is the total water loss, and this is the stomatal conductance. So this enables us to see this very interesting strategy of this resistant plant, where we know from the field this plant produces much more under drought. Now, remember, this is the average of 4 to 5 or even 6 plants. How can we get such a nice resolution even at a very low transpiration? You see the plant at this point transpire relatively low amount of water. It's in the gram level. And we have everything in a noisy environment. So one of the big things that PlantArray knows to do is to select the signal from the noise. Okay? What is the noise? The noise is temperature. You know, we are in a greenhouse, so there is a lot of fluctuation of temperature. The noise is long cable. If you work with lazy meters, with cables and data logger, like the old systems, the cables take a lot of static noise. The noise could be vibration. Just if somebody walk in the greenhouse, you have noise. Noise could be drainage from the pot. Noise could be evaporation from the soil. So PlantArray knows how to take all of these noises, filter it out, so you get a very clean signal that enables you to see this result that I show you. Just to show you what are these problems that I mentioned here, I want to show you because it's very hard to understand until you see this noise. For example, you see this is normal pot that actually drip on the lazy meter. You see the water in the bottom. This is something you cannot control in the normal lazy meter system, and you just lose this water that later on evaporates. Also water that evaporates from the surface. So people put this plastic bag or use these caps underneath, these plates underneath. You can see it here. But this caps doesn't help because water is still going to evaporate from here. And moreover, after a few days, you're going to get algae in the water, as you see in this system here. This algae itself brings you some noise. So this cannot be done if you don't want to have noise. And of course, you can see this spot here. You see these spots here. These are water that was evaporated just because it's a greenhouse. And also this concrete floor actually transpire vibration to these lazy meters simultaneously. So this old-fashioned lazy meter cannot help you. What Plant-Ditech has a lot of this gadget. Actually, it has 2 patents on this, covers, O-rings that you see here, this green bath, and many other systems that enable you to filter out the noise directly to the controller, which is very close to the system, which is a very big part of selecting the noise out. And this is how you can get very nice signal to noise. Okay, this is the filtration of this funny looking bass and rings and stuff that you have here. And also the system enables you to separate between the soil, the root, and the shoot, and the water in the soil. So this is how you can really get a clear transpiration, a root flux, the absolute water in the soil, and to know exactly how much water went in and went out because nothing is dripped away. Going back to our story, so we did this screening to all of our lines. I'm not going to get too much to details. We later confirmed that the stomatal density and aperture, we actually did it with running in the field, many people running in the field and take the imprints of the stomata 8 times a day just to see that the PlantArray is correct. And we found that the plant with a very nice performance had much more stomata on the bottom part. You see on the adaxial part, they had much bigger stomatal density. You see like this 5.2. Then the 18.1 that had similar amount of stomata on the top and on the bottom part. Also, the 18.1 open stomata at the middle of the day. You see around 12, this plant opens stomata to be the widest, while the 5.2 open it much earlier, as I showed before from the transpiration rate. Now you can also see it in the stomatal aperture. This is the actual stomatal aperture measurement. So we can actually confirm what we measure in the lazy meters and in actual measurement. I'm going to skip the genetics, but we find very nice correlation also to some gene that express there. This, by the way, if you want to see this paper, is now on the bioRxiv, and I will be glad to send you a copy if you want. To summarize what we found, so high transpiration is this is comparing the very high productive to the low productive. So fast grow and high transpiration at the early growth is a very good trait, functional trait. Fast closing of stomata under drought is a very good trait. You can see this here. Having more stomata on the lower part, on the adaxial part, it's a very important part. Having this golden hour, so open of stomata early in the morning and close it at noon is a very good stress-related phenotype. And also a very fast recovery. So all of these traits, functional traits that I mentioned here, was found to be a very important physiological trait for drought response. I'm sorry that I'm rushing because I want to show you two more examples of the use of the plant array. One of them was just published in Plant Physiology, where we could measure a functional trait of a cellular level. And let me explain what we did here shortly. So what we did here, we measured the permeability of water from the spongy mesophyll. As you can see it here, we could label them in GFP and we can extract the cells and measure the water permeability. So we know the different water permeability of the pellicide cells, the spongy, and the bundle sheet, each one of them separately. What we found is that the mesophyll, the spongy mesophyll, and the bundle sheet has much better water permeability than the pellicide. But surprisingly, we could not get it with the lichor. With the lichor, we get very high water conductivity, water transpiration, much better than the wild type, as expected. But we could not distinguish these two lines, although we knew we should have a difference. Why is that? Because the lichor, even if it's a very sophisticated machine, but you can see that when you try to regulate the VPD in the chamber, if the plant transpires so much like our lines, the machine has a very hard time to do it. So in the end, you get some kind of bias in this chamber, unless you put a very strong wind speed, which gives you another bias. Another thing is this article published in Nature Plants recently, where Farquhar, the original scientist that actually wrote all the algorithm for the lichor, tells us that recently he found that there might be some problems with the original calculation, assumed 97% humidity in the substomatal cavity, and today he showed that it might be down to 80. This is a huge difference. Anyway, we could not see our phenotype in the lichor, so we went to the lysimeters, and this is all we could do this data only because our filtering ability of the Plant-Ditech. Think about it. The Plant-Ditech can give us this very nice graph coming from Arabidopsis. It's a very small plant. So we could see the Arabidopsis plant transpiring on the lysimeters, and we could see the differences we expected to see. Here you can see a very nice differences between the different line. And the reason I'm telling you this, the only way to see these nice differences in Arabidopsis is you have only if you have a very good signal to noise ratio. In the last minute, I'm going very quickly to tell you about a biostimulant experiment we did. Again, biostimulant, you can do it, of course, on crop plants, so the size of the plant is not a matter. But here, the stimulant sometimes work and sometimes it doesn't work. And when it doesn't work, you don't know, was it my measurement or my plant or the time? So measuring continuously and simultaneously all the plant together give you a big benefit as long as you can reduce the noise. In this experiment, it was published, so I can send you the paper if you want, no problem. We did 6 different treatments with commercial biostimulant, it's called seaweed, a new biostimulant developed by ICL company, and just control normal irrigation. And we put them under normal condition and drought just to save the time. I will show you the final result. What we saw in phase 1, the seaweed have a very big advantage on the plant growth. So the seaweed had a very nice phenotypic advantage of fast growing, but these plants grow fast but recover to drought not so good because they started the drought at a bigger size. And the fact was that they actually recover slower, although they were treated with this seaweed. But this new chemical that was actually a mixture of polysugars and amino acid, okay, this mixture of this, what we call the new biostimulant, improve a bit the plant vigor under normal condition, but improve a lot the plant response under drought. And the result was very nice resilience, which means recovery from stress. Okay, I think I overdo my time, so later on I will be very happy to answer the questions. Thank you.
Thank you very much, Menachem. Very interesting. I hope that we can have Karen back. I think, let me put her again as a presenter. There was a problem with her computer. Let me. Okay, Karen, would you like to try and turn on your camera now? Perhaps it should work.
Yes, here I am.
Thank you so much. Thank you both for these very, very interesting presentations. I think we can now take questions from the audience. There was one question I noticed in the chat. I'm going to start reading that. It was from Emilio, I think, I noticed. He was asking what kind of sensors is the PlantArray based on? So that was the first question we had in the chat. I think probably it was some detail that went missing. Thank you.
Yes, so I can answer that. So the main sensor we're using is the weight or the scale load cell sensor, which is a very sensitive sensor for weighing the weight of the whole setup. And of course, from that we extract the specifics about the plant. Also we use atmospheric station and sensors in order to understand different characteristics of the atmosphere dynamic. We take the measurement the same time of the weight together with the atmospheric sensing. Atmospheric station includes several parameters of atmospheric sensing, like light, humidity, temperature, and we extract the VPD from that. So these are the basic, but we also have additional atmospheric sensors, for example, sensing like wind or soil sensors of different kinds for EC, for temperature, for water content, and pH and others. And also we have plant sensors, additional plant sensors like leaf temperature and NDVI. And we also have in the solution for the fertigation, we can also add different sensors there. And in general, we work with a certain communication mode which allows to add more sensors. So if there is a need, it's a common communication code, so it can work with additional sensors that also maybe you are using or you want to add to the system.
Thank you, Karen. I see more questions in the chat, so I will read them in order. I guess they're all very much about how you measure different things. So the next question was, how do you estimate GS? So it's that stomatal conductance, I guess. I don't know who of you would like to take this question, perhaps Menachem?
Yeah, gladly. I just want to add on the sensor part that one of the nice things in the sensor, you just plug them in. You don't need to calibrate them, and immediately you get your units. For example, if you put your soil sensor, you can get the soil water content if you have the right equation. So this saves a lot of time in the work. Yeah, regarding the stomatal conductance. So stomatal conductance is basically the water the plant loses divided by the VPD divided by the plant weight. So if you take a like or gas exchange mechanism, so it does the same. It takes the water flux, really water flux, Lycor takes it in millimole per minute based on infrared measurement. We take it based on the weight. We see the weight change in every minute. And then we normalize to the whole plant biomass because we take the whole plant and we give absolute value of the whole plant. And so basically, dividing the transpiration per mass per VPD. The VPD is the delta of the water potential. This is pretty similar of what Lycro did. Always GS is a calculation. Also in the gas exchange, it's never a measurement. It's always calculation taking the flux of water on the pressure of the atmosphere divided to some area or weight. By the way, the Wageningen University test our model and publish it. We got 99 point something accuracy compared to a porometer.
Thank you, Menachem. There's a couple of questions from Lorenzo. One is, he was asking, can you better explain how the system is able to apply the same drought steps for all the plants? So you mentioned that we just don't just close the tap, but how can you basically tailor the stress to the different plants so that you apply a kind of equivalent type of stress?
Yes, so we use a specific feedback irrigation algorithm which measure the plant transpiration. For example, one plant transpire 200 ml per day and the other one 100 ml per day. And you want to give both of them similar drought. So we can give the 100 ml per day 50 ml and the 200 ml per day 150 ml. So both will get, it's just an example. So this example, you can just reduce the water based on the transpiration. Another way is to use a soil sensor and get a feedback irrigation based on the soil water content and tell the system, keep it in a similar soil water content based on the soil sensor. Another way is just irrigating based on a target weight. So you can say, okay, I want to keep all my plants on this target weight. So the system will irrigate all of them on the same target weight. So there are several scenarios. I like the most the dehydration rate. So not keeping them on a fixed level, because this is, to my belief, something you don't get in the field. What you do get in the field is dehydration. The soil is gradually drying, and you try to mimic this slope. So we try to calculate what is the slope rate we want. By the way, if we are not sure, we many times do a preliminary experiment doing 2 or 3 different kind of drought, fast one, middle one, and slow one, and then decide how to conduct the drought. But it's a very flexible because each plant has its own irrigation and its own data from the plant. So it's just a feedback.
Thank you. There's a couple of questions also from 2 different people about how you calculate biomass and how you separate the weight loss due to transpiration and weight increase due to biomass accumulation. So if you, I guess, Menachem again, can you answer this?
Yes, sure, gladly. So the system has several plastic gadgets. One of the most important is this green bus that you saw. This bus enables us to know exactly how much water get in and how much get out, because we also allowed leaching, washing of the soil. We also have a mesh in the bottom of the plant that doesn't allow any sand to go, any sand at all to go. And we can irrigate the system always back to the soil water or field capacity level. That means that after, let's say, every round of irrigation, when I bring my plants to this field capacity, the weight is plus minus 2-3 grams, does not change. Even if it's a 5 kilo pot, it does not change, because after the last drop leave the plant, the soil hold the water and all the plastic stays the same. But when you know to get to this field capacity very accurately, and you irrigate the system like this, and then the plants start to grow. So instead of getting the same weight as you expected, the system start to gain weight. This gain of weight is coming only from the plant. And this gain of weight coming under normal irrigation conditions. And this is relatively easy. What happened under drought? Under drought, you cannot get field capacity, of course, because you want to get drought. In this scenario, we have a calculation algorithm that study the plant before the stress start. So the algorithm study how much water the plant lost to how much gram the plant gained. And based on this water use efficiency slope, the system can calculate the plant gain based on the transpiration. Transpiration always be calculated very easy because this is only the scale measurement. So the first few days of drought, we can get a very accurate estimation of the weight. If you keep it for a very long drought, then you can start losing the accuracy of the weight. But for normal irrigation and the initial drought, it's pretty much accurate.
Thank you. There is a question from one of the audience that I guess perhaps he can explain better, or she, I don't know. Also in India, scientists are interested to carry out such phenotyping for seeds. Do we have a workflow for seeds? I don't know if you, Ganapati, would like to address this question directly to the speakers. I'm not sure I understood completely your question.
No, I can understand. Maybe I'll start and Menachem can complete my answer.
Okay, thank you.
Because he did some trials with that. But I can say what we do normally. Normally, what we direct our customers is to do whatever treatments they need to do for the seeds, and then plant the seeds and get them to a level of where you have plants to heights of about 10-15 centimeters. This is more or less the good starting point to start the experiment because they gain enough weight. And then from that point to start the experiment and do the analysis. But I know Menachem did some trials. Maybe Menachem, you have some other ideas?
I'm not sure what they mean by seeds. So seed per se, we cannot measure. But if they mean seedlings or seed treatment, if it's seed treatment, no problem. You do the treatment for the seeds and then you measure the small seedlings. If it's seedling, we have two approaches. One is to put many in one pot. It's not a problem. So we can put like 20 or 30 seedlings, and then the transpiration is very nice. Like I show you with Arabidopsis. For example, if you do wheat or barley, you can put 20, 30 seedlings, and up to the age of 2 to 3 weeks, you get the bush, and then you have the measurement of a very young age. But if you meant seedling in a chamber, like they sitting in their own small pots, we also have a solution, but it's a pretty much man-made. It's not coming directly from the system. We use the system capability, but it's less accurate. So I'm not sure what the audience asked, but this is what we do in seeds.
Perhaps, Ganapati, you can get in touch with the speakers to further develop this conversation. I have a couple last questions before probably we say goodbye at 5 o'clock. There are two different questions about the possibility of adding cameras or multispectral or RGB, some imaging phenotyping to be integrated with the system.
Yeah, so we have a system that we have cameras on top. We can work with any supplier that supply camera. The system is open for upload and download of information either by CSV file or automatically with an API. So we can work basically with any type of camera, and we have several of those already in our customer base integrated together.
Also, the system has its own NDVI, PRI, and infrared built-in. It's not a camera, it's a sensor, which gives you a continuous measurement. So if it's NDVI, PRI, or thermal, you can just put a sensor directly to the plant, and then you get this advantage of continuous and simultaneous measurement. We also have at least one or even two publications with hyperspectral camera on top of the system that was published in the last 2 years.
Yeah, there are some publications with this kind of integration. Yeah.
Great. All the publications are available on your website anyway, so you know the audience can then find more information in the literature there. I'm not sure there are any other questions. I don't see other questions in the chat. I hope I haven't missed any questions, but I guess your contacts are available. Ganapati, you wanted to comment about the seeds?
Yeah, am I audible?
We can hear you. Yep.
Yeah. Okay. See, now off late in India, this phenotyping is picking up like anything. And last week, when I was in one of the most prestigious universities, Tamil Nadu Agricultural University. They were asking about similar kind of phenotyping if it can be done for seed, and they are more interested in it. So any comments on that?
Well, I know there are some other solutions available for screening seeds. For us, it's not, let's say, the seed by itself is not let's say, the target audience in terms of phenotyping. But as I said, we can do seeds in terms of whole plant measurement. So after you start growing the seed, we can do the phenotyping. So whatever you do treatments, or you want to know the phenotypes of the whole plant, but you have to grow it for a certain level in order to do that.
Thank you. Thank you for your question. I think we can wrap up. I would like to thank again our speakers today, Karen and Menachem. Thank you so much for sharing your knowledge in this very interesting presentation. I remind the audience that the recording will be made available on the Plant-Ditech website. And thank everyone for attending. Thank you.
Thank you, Laura. Thank you, Laura, for doing the hosting for this seminar. Thank you so much.
Thank you, guys.
Thank
Goodbye.
You.